All right, good morning, and thank you, Kieran, for the kind introduction, and thank you to you and your colleagues for putting this important and timely workshop together. And I certainly hope that each of the participants will gain valuable knowledge and insights from what they learned from the workshop today. So we had the privilege to have Kieran here as our keynote speaker in Florida a couple years ago for a graduate student symposium. It was great to hear from him, and it's great to set up collaborations with him. And it's certainly my pleasure and my honor to speak to you guys today about a topic I'm passionate about, vitamin D, cattle, and immunity. So before I go further into my talk, I want to address the issue of the relevance of cattle, vitamin D, and One Health. How do they all relate? So why cattle, why vitamin D, and how do they relate to One Health? And first, cattle are a critical part of sustainable food systems, but also perhaps the most controversial part of sustainable food systems. And shown here is a picture of one of our cattle farms in Florida. And while the cattle farms are often criticized as being contributors to climate change, lost biodiversity, and decreased soil and water health, amongst other issues, I'll argue that if we continue to improve management to address the environmental issues of cattle, that they present, they will continue to be a vital part of sustainable food systems. And the reason for this is depicted in this photo is that cattle, as a ruminant species, convert forages such as grasses that do not have nutritional value for people into a very nutritious food product such as meat or milk. Now, I haven't yet visited Ireland to see the beautiful pasture lands there, but I've seen pictures and heard about the beautiful and valuable pasture land there in Ireland. And that asset that you have there, that's one of the great assets of what cattle can take there and convert that into a usable and highly nutritious food. However, there are several issues that threaten the sustainability of cattle. Amongst these are diseases of cattle, particularly the most problematic yet here in the US anyways are mastitis and metritis, inflammatory diseases of the mammary glands and of the uterus. And so these are typically caused by bacterial infections. They also result in decreased milk yield and decreased fertility of cattle. They're also a reason for the greatest usage of antimicrobials. So not only are they affecting productivity of cattle, our current reliance on antimicrobials to treat these diseases is not sustainable because of the emergence of antimicrobial resistance. And a third point about the diseases of cattle and their importance is that the threat of zoonotic diseases such as tuberculosis. Now, the imminence of these zoonotic diseases isn't quite what it once was in developed countries such as the US and Europe because there's fewer people working around cattle, but it certainly is still a threat, and on a global scale, this is a major issue. And the final point I'll make here is why study vitamin D in cattle. It's a useful model, as I hope you'll see from my data that I showed today, to understand vitamin D physiology and nutritional requirements, not only for a species that has relevance for our food systems, but also to understand the vitamin D physiology and nutritional requirements for the human population. So I'll give you a brief overview of what I'm going to present to you here today. So shown here is a simple schematic of vitamin D's actions in the body. On the right is the classic mode of action of vitamin D in the endocrine system, where it functions primarily to regulate calcium and phosphorus homeostasis. And we still do a considerable amount of research to study how that functions in cattle and the relevance of that. And that's contributed, the studies that have been conducted in cattle have contributed to much of what's known about vitamin D metabolism now today. I'll focus most of my time on the figure on the left here on the actions of vitamin D in the immune system. That's something that I've studied since I began my PhD work several years back, and I still continue to study to this day. So I'll present evidence on the existence of this vitamin D endocrine system and the bovine immune system. I also present some of the implications this has for mammary immunity and mastitis prevention. I'll spend most of my time presenting some more recent data on the effects of vitamin D status and dietary vitamin D treatments on health and productivity of dairy cattle. Before I go on, one thing, a note just on nomenclature. Up in the upper left here, I present the vitamin D pathway. And you've all probably seen these terms used at some point already today, but I just want to make sure everybody's clear on what I refer to. So vitamin D collectively refers to any one of these corticosteroid molecules up here. Shown are the vitamin D3, would also refer to vitamin D2 as well. Vitamin D3 in cattle is the primary metabolite in cattle, just as in humans. The second metabolite is the 25-hydroxyvitamin D3, which I'll commonly refer to as 25D, which is then readily converted to, or through the 1-hydroxylase is converted to the active metabolite 1,25-dihydroxyvitamin D3, also known as calcitriol. So shown here is the model of what I've put together on how vitamin D is believed to be working in the bovine macrophage. And for those of you that have studied how this functions in the human macrophage, it's very similar in regards to its activation. There's some subtle differences in the proteins or genes that are regulated by vitamin D, but in general, the outcomes are quite similar. So in this endocrine vitamin D pathway, toll-like receptors or other pathogen recognition receptors, along with cytokine receptors, will stimulate the induction of CYP27B1. And that's what's shown here in this panel in the middle, is the effects of various stimulants on CYP27B1, the 1-hydroxylase, the gene for the 1-hydroxylase in bovine monocytes. And so the induction of CYP27B1 is critical because it allows for conversion of the 25-hydroxy-D to the active form, the 125D, within the monocyte. So the outcomes of that, there's several that we've documented in the bovine monocyte, and they would include induction of antimicrobial proteins such as inducible nitric oxide synthase, beta defensins, antioxidant proteins such as the thyrodoxin system and metallothioneins, and several chemokines and cytokines that are induced by 125D in the bovine monocyte. So one of the functional outcomes of this is increased antimicrobial activity. And that's what's shown here by this in a paper published by Garcia and colleagues at the National University of Mexico. And here they treated bovine macrophages with 125D and observed an improvement in our inhibition of Mycobacteria bovis intracellular growth by treatment of bovine macrophages with 125D. So this is work that's been done in vitro. We've also studied this mechanism or this pathway in vivo, and that has considerable implications for mammary health of dairy cows. And this is one of the udder of dairy cows is a prime site for bacterial infections, and mastitis in cows is one of the major uses of antimicrobials. So this is a key area of interest for us to curb the use of antimicrobials. And so we've studied this, the activation of the vitamin D pathway in the mammary gland during mastitis. Here's data shown from cows where we induce mastitis using an intramammary LPS infusion. And then we measure the expression of genes of the CYP27B1 and the vitamin D receptor after the intramammary challenge. So you can see from this that there's a very rapid and robust increase in both of these proteins. Not shown here, we've also demonstrated that the immune cells in the mammary gland are able to respond to vitamin D ligands, either the 25D or the 125D from intramammary treatments. So that these cells do in fact respond to the increased 125D production during an intramammary bacterial infection. So there are opportunities that we have to apply vitamin D therapeutics for intramammary treatments, either as an adjunctive or as an alternative to current antimicrobials that are used. And that's one area that we have been working on. A second area that we've been working on is the vitamin D nutrition of dairy cows. So when we started this work a few years back, we started to ask the question, well, what is the current status of vitamin D in cows in the US? And we started sampling herds across the US. And here's a compilation of data. This is from nearly 1,000 animals, mature cows, that would either be supplemented or be out on summer pasture, so they'd be exposed to sunlight. So this distribution of vitamin D, the serum 25D, amongst the population that we've studied is certainly a bit different from the human population, where it's going to be skewed a bit more to the left in the human population. Just to put this scale in perspective, I know earlier talks had this presented in nanomoles for serum 25D. So if you just multiply this by 2.5 here, you'd have a range of 100 to 250 nanomoles for serum 25D. This red dashed line here is what's been proposed as the indicator for insufficiency in cattle. And you can see there's not many cows that would be considered below that line of insufficiency. And there's a couple of reasons for this. For dairy cows in the US, they are generally supplemented with vitamin D at a rate of about 1 microgram per kilo. I should say 1 microgram per kilogram of body weight. The amount that I have there is incorrect. It's 1 microgram per kilogram body weight up to about 1.5 micrograms per kilogram of body weight that cows typically receive, because supplemental vitamin D3 is relatively inexpensive, especially compared to other vitamins such as vitamin A and vitamin E. So vitamin D deficiency is generally not a problem in cows. However, when we start to break this across different regions, we start to learn more about factors contributing to vitamin D status. So I took this population here that's on the left and just broke this into various regions or conditions that cows may be in. And so here is you would be different than what you might expect. Cows in the southern region are actually somewhat lower than we found in cows in the northern region. And this is largely due to the fact of again, vitamin D is oftentimes supplemented for our dairy cows here in the US. And I'll point out that these cows here that are below this threshold, the 30 nanogram per mil threshold that we set, those cows have been receiving just below what is typically recommended for cows. So they're receiving around 0.5 to 0.75 micrograms per kilogram body weight, whereas most of the animals here that would be in the northern regions that we had sampled, all of these herds were receiving somewhere between 1 to 1.5 micrograms per kilogram body weight. So in comparison, you see cows here that would be on summer pasture. They generally range from about 40 to 90 nanograms per mil for serum 25D. Now I put this data in here from Ireland. This is data that Kieran had shared with me a couple of years back. These are from dairy cows that were sampled in the springtime. And the key difference here is that in Ireland and Europe in general, vitamin D is not supplemented at quite the same rate we do for dairy cows in the US. One particular reason for that is there's much more strict guidelines on inclusion of supplemental vitamin D in animal feeds in Europe than what there is in the US. We actually don't have any regulatory limits for supplemental vitamin D in animal feeds here in the US. So here's certainly an opportunity within Europe and Ireland in particular to examine, is this contributing to animal health problems within the region there. So one other thing I did is break this into differences of lactation stage. And so one thing that's we observed in this data set, plus others have published this elsewhere, is that cows in early lactation have lower vitamin D status than cows in either late lactation, or if you compare them prior to giving birth, the cows in early lactation here, just after giving birth, have a lower vitamin D status. And this has been something shown in several different data sets now. Furthermore, within this group here of early lactation has been demonstrated with some recent work out of Michigan State University that cows that have a lower serum 25D compared with cows with a greater serum 25D are at greater risk for uterine diseases such as mastitis and retained placenta, ultimately resulting in decreased fertility of animals. So we do have an opportunity here to examine what's the effect of that low vitamin D status. Can we improve that? One other area that we've also explored or observed a difference in vitamin D status would be the young calf. And so this is pointed out earlier and for human babies, is that at birth, vitamin D status is very low. And we see the same thing in calves. And it's just like in humans, the vitamin D status of the calf is dependent on the dam. And so you can see a rise over time here. So this would be these would be calves born in spring, and this is by the end of summer, they have a sufficient vitamin D status. But there's many animals that are very low in vitamin D early in life. So this presents some opportunities for vitamin D intervention for us. One, we can target the postpartum cows because they have increased risk for disease. And there's data showing that vitamin D status is associated with uterine diseases in those cows. We also can target vitamin D nutrition for calves. The young calf is at greatest risk of morbidity and mortality within the first few weeks of life. And calves are also, as we showed in the previous slide, born with low vitamin D status. So we've begun doing experiments in the last few years testing the effects of vitamin D source and the amount of vitamin D on vitamin D status of cows. So when I say we test the effects of source, we've been comparing calcidiol here, the 25D, to supplemental vitamin D3, which is the normally supplemented vitamin D for cows. And what we found from that is the 25D, the supplemental 25D, is much more effective at increasing serum 25D compared with the normal vitamin D3 or the cholecalciferol. So here in the filled symbols here, the black symbols, these are cows supplemented with either 1 or 3 milligrams per day of the 25D, versus here cows that are supplemented with either 1 or 3 milligrams per day of cholecalciferol. You can see there's no increase here between the different levels of cholecalciferol. And this is not necessarily a problem with absorption in cows. It's actually a problem of converting the vitamin D3 to the 25D3 in the liver. So we've also done, so the data set on the left is from lactating dairy cows. The data that I show here on the right is from young dairy calves, from bull calves that we tested this in. So we very similarly tested either cholecalciferol at 1.5 micrograms per kilogram body weight versus 3 micrograms per kilogram body weight versus similar levels of calcidiol. And so we again see that there's supplementing the 25D or the calcidiol is much more effective than the cholecalciferol. And here's the plot on the right, just shows the response according to the dose in micrograms per kilogram of body weight. So again, the calcidiol is a much more effective route at increase in serum 25D. So one thing I will point out for the dairy cows over here on the left, I'll go back to that again. So if you're paying attention to the levels I showed earlier, these animals here would be considered sufficient. They're at about 50 to 60 nanograms per mil of serum 25D. And we're increasing these to well over 100 or even up to over 200 nanograms per mil. These levels at a short term are not toxic, and I'll show you have been seen to be beneficial, but in the experiments that I show here, we're not necessarily comparing vitamin D insufficiency to sufficiency. We're comparing vitamin D sufficiency to elevated or super physiological concentrations of 25D in serum. So we've conducted several experiments now looking at the effects of vitamin D source on health and production of cows. In this experiment, the data I'm showing here from a series of papers published by Natalie Martinez at the University of Florida in the Journal of Dairy Science, we looked at the effect of feeding 3 milligrams of 25D versus 3 milligrams of vitamin D3 in late gestation. And from that, we observed that cows had increased milk yield, increased colostrum IgG, which is very critical for passive transfer of immunity. We saw cows had decreased retained placenta and decreased incidence of metritis when fed the 25D versus the vitamin D3. And so going back to what I showed earlier, the effects of vitamin D and immunity, when in a second experiment, we tested either vitamin D3, the cholecalciferol versus the calcidiol at 1 or 3 milligrams per day, and then measure immune cell gene expression of peripheral blood leukocytes, we can see that there's a number of genes that are affected that have relevance to either cytokine production, pathogen recognition receptors, beta defensins, that are all, or chemokines that are all upregulated in response to feeding the calcidiol or the 25D versus the regular vitamin D3. Similarly, we've found that feeding calcidiol at 3 milligrams per day versus the normal vitamin D3, the cholecalciferol 1 milligram per day, there's a decrease in mastitis severity. So here's cows we've given an intramammary strep uterus challenge to induce mastitis, and the cows that were fed the 3 milligrams per day of the 25D had a decreased severity of mastitis. Those cows also had increased presence of neutrophils in the milk of the infected glands. Neutrophils are one of the key cell types responsible for combating the infection. So the last data points I'll show here are then the effects of maternal vitamin D on vitamin D status of offspring. And there's some of this work that was shown for the human population, one of your talks earlier. And we see that very much the same thing taking place in cattle, where the newborn calf is dependent on the vitamin D status of the mother. And here's an experiment where we fed gestating cows either 1 milligram per day of cholecalciferol or 1 milligram per day of calcidiol. And this is the vitamin D status then of the calves that were born to those cows. And so the control cows here received just a minimal vitamin D supplement. And you can see there's an increase here in vitamin D status for those calves from either the cholecalciferol or the calcidiol supplemented cows. And this has implications then for the immune status of the calves. This is monocyte CD14 expression here shown on the right. And you can see there's an increase in monocyte CD14 with the increase in serum 25D status of those calves. So just like you've seen in the human population, maternal vitamin D supplementation is also critical for vitamin D status of the young calf. So with that, I'll conclude with these points. What I've shown here today, again, and demonstrated in this figure here, vitamin D contributes to functions of the bovine immune system, in particular antimicrobial, antioxidant, and chemokine pathways that we've shown. We've also, from surveys of vitamin D status among herds, indicate there's potential for vitamin D interventions in peripartum cows and neonatal calves. And finally, we've shown that dietary vitamin D improves health outcomes and indicators of immune defense in cows. So with that, thank you to certainly all the students that have contributed to this work, my collaborators that have been excellent, and faculty here at the University of Florida that have been great colleagues. Any questions that you may have, because I won't be at the discussion panel later today, feel free to send me an email at cdnelson@ufl.edu.